Okay, students, today we will be talking about the principles of cell signaling. So I'll just first introduce you to cell signaling and signal transduction. So signal transduction refers to any process by which a cell converts one kind of signal or stimulus into another by an ordered series of bioreactions inside the. Now we know that eukaryotes are characterized by multicellularity, where or a living system is dependent upon the coordination between cells. Right? The living system can work properly only coordination between cells. Cells are autonomous, but these systems are dependent upon each other for some of the proteins, some of the ligands that can allow a particular organ to function properly. So, cell to cell signaling basically can be understood as, say, for example, we'll take the example of here, an endocrine produces a hormone that travels through the bloodstream initiates a biochemical reaction or a physiological response in another called its target cell. So this is a very simple example of cell-to-cell communication and cell-to-cell signaling. And that signal is said to be transduced because that signal is produced by you can say distinct cell, which may be distant from the target site, but it can travel or flow through the bloodstream, reach its target site, and initiate a biochemical reaction inside it. Now, these secreted molecules, which are the signaling molecules, mediate via different forms of signaling. Now, ahead in this lecture, we'll see there are different types of signaling also. Signaling molecules that a cell secretes may be carried far afield to act on distant targets, or they may act as local. That is, a cell can produce a signal of its own also, affecting only cells in the immediate environment of the signaling. Now, multicellular organisms are biochemical messengers, the ligands, for example, the hormones. Now, signaling can be local. In local signaling, cells may communicate by direct contact or cell-to-cell recognition also. You may be aware of different cells between cells. That cells can communicate to each other by direct or cell-to-cell contact also. Then, if we take the example of the cells of the immune system, they can, they can travel to distant tissues where cell-to-cell recognition is important, where they understand what is the site of infection or what is the where in which tissue that infection is present. They recognize them due to changes in the you can say cell surface behavior of that infected tissue, and basically also example local. Now, in many other cases, animals communicate using regulators, messenger molecules that travel only in certain directions. So that is, in case of local signaling, you see that the the target cell not vary. Then, in comparison to local signaling, if we talk of long distance signaling, where there you will see that plants and animals use chemical messengers called hormones, and that can travel long distances. And the ability of a cell to respond depends on whether or not it has a receptor specific to that cell. Now, when cells are very near to each other, in that case, it is not difficult for the signal to reach its target site, right? Now, if we talk of long distance, and suppose the flowing through blood, it might travel to, or come in contact with different tissues. So it actually binds to target site, and it has the receptor for that particular hormone or that signaling molecule. So this is what is point onto a signal completely depends whether or not it has a receptor specific to that signal. That is, it has that cell surface protein or that can bind to that signal and initiate a biochemical reaction in the cell. So in this figure, you can see local signaling that is explained here, where you can see the target cells are very near to each other. The very common example you can see of nerve transmission, okay, through synapse, where you see these neurons, they extend to long distances, but there is a small gap between the nerve ending and the so these are examples of local signaling, whereas the endocrine signaling, as I had explained in the previous slide, is the prominent example of the long distance signaling. Okay, so it is also called signaling. So the endocrine gland, it produces the hormone through blood vessels. It travels to the target site, which may be very distant in the system, and brings about or initiates a biochemical reaction in that particular cell. Now, let's talk of the types of signaling. Now, signaling is basically classified into three types. First one is autocrine signaling, second is paracrine, and then comes the endocrine signaling. So, first we'll talk of autocrine signaling. Okay, auto, as the name implies, denotes self. So in autocrine signaling, cells respond to substances or chemical messengers which they themselves release, and thus changes takes place in the cell itself. That is the cell that is actually producing the signaling molecule or the ligand. I'll just show you in this figure. Here you can see this is the cell which is actually releasing the you can say signaling molecule. I should not say a hormone because hormones normally travel long distances. And then you can see in this green color, these are the receptors which are present on the cell membrane of this particular cell. So it has target sites or receptors for this particular ligand on its surface. And these receptors basically they initiate a response in this particular cell. And here in this diagram, in this next part of this figure, you can see that a number of cells, when they produce because a tissue can contain a number of similar type of cells, the number of signaling ligands will be more and the response will be fast. So a single signaling cell receives weak autocrine signal, whereas in a group of identical signaling cells, each cell receives a strong autocrine signal. So a cell secretes a hormone or chemical messenger that stimulates its own growth and proliferation. Autocrine signaling is a characteristic feature of tumor cells, many of which overproduce and release growth factors that stimulate inappropriate and unregulated cell proliferation, as well as influencing adjacent non-tumor cells, lead to formation of tumor mass. So this is the best example. In case of there is uncontrolled multiple cells, it is because a cell can produce go and bind to the receptors on their own cell surface and bring about cell division. In autocrine signaling, a group of identical cells produce a higher concentration of secreted signal than does a single cell. This we have in this particular figure. So this is autocrine signaling, means the cell which is actually producing the signaling molecule, it goes and binds to receptor on the same cell and brings about a change in that particular cell only. Next, paracrine signaling. Now, para means near. So, in paracrine signaling, the signaling molecules or ligands released by a signal releasing cell, which is called the secretory cell. If we talk about only those target cells which are included in the proper target, the secreted signaling molecules must not be allowed to diffuse far. For that, they are often rapidly taken up, destroyed by the extracellular enzymes or immobilized by extracellular matrix. Now you know the specific. And response to a signal by a cell cannot last forever. I mean, it must be for some fraction of time, say, for example, muscle contraction. So, any cell that is a paracrine signal that has to act on nearby cells, what it does is after the release of that particular ligand or that signaling molecule, it is to be made sure that it should not diffuse too far. So, I mean, after a particular frame of time, either that ligand is taken up by nearby cells. If suppose all the receptors have been occupied by the ligand and still there are portions of that ligand that are left in that gap between the two cells, they are destroyed by the extracellular enzymes or they are immobilized or you can say trapped by certain components of the extracellular matrix so that the response is ended there and the target cell is not overstimulated. The conduction by a neurotransmitter one cell to another or from a nerve cell to a muscle cells occurs via paracrine signaling. So, the signaling at the synapse, which occurs with the help of neurotransmitters, is the best example of paracrine signaling that you have seen in the first diagram shown you in the beginning of the lecture. Here you can see the diagram of paracrine signaling or this figure representing paracrine signaling. This is the secretory cell and this is the adjacent target cell. The ligands released by the secretory cell are taken up by the nearby cells. That stimulus is taken up by the nearby cells through cell surface receptors. And again, here you can see this is the signaling cell and this is the local mediator that targets the nearby cells. Now let's move to endocrine. In endocrine signaling, the signaling molecules, that is the ligands or the hormones, are synthesized and secreted by endocrine cells, transported through the circulatory system or the tissue fluid of the organism, and finally act on the target cells distant from the site of synthesis. Endocrine signals are called hormones. The target cells have receptors for binding specific hormones and thereby pull the appropriate hormones from the extracellular fluid. Endocrine hormones, for example, insulin and epinephrine, are synthesized and released in the bloodstream by specialized ductless endocrine glands. You all have learned the endocrine system. You have learned about the different hormones, their, their action and their target cycles of endocrine signaling. So here you can see the figure of endocrine signaling where the hormones produced by the endocrine glands through blood vessels, they can act on different target sites which are having receptors for hormones. Now, after learning about the types of signaling, let's learn about the mechanism of signals transduction or what are the basic steps in cell-to-cell signaling. Reception, transduction, and response. Now we'll see what are they. Now just take a look at you can see here this light brown part which represents the cytoplasm. This is the cell membrane. In this orange color band, you can see a plasma membrane, and on this membrane is present this transmembrane protein. Because it crosses both the leaflets, that is the complete lipid bilayer. It has extracellular side that is facing the cell exterior, and an side that is facing the cytoplasm. So reception means that the signaling molecule, say for example, this is an endogland, say produced by any, you can say endocrine gland. This is a hormone produced, and this signaling molecule will travel and go and bind to the extracellular side of this receptor. So this part is called reception, where the ligand actually binds to a receptor on the target site. So receiving the signal on the cell surface denotes the reception part of signal transduction. Okay, then next comes the transduction part. Now, once the signal goes and binds to the extracellular side of this transmembrane protein, that is the receptor, this stimulus is transmitted across the lipid bilayer through the transmembrane domain of this transmembrane protein, and the it induces a change. It can be a canal change, or you can say if the intracellular site acts as a, you can say, enzyme, it can lead to production of some intracellular molecule, and the message that the ligand is there, what is the type of ligand, and what actually biochemical reaction has to be initiated in the inside the cell. So this message is passed through the transmembrane protein towards the intracellular side into the cytosol. This is called transduction. That is initiation of the biochemical reaction. So you'll see that during this process, a number of biochemical reactions will learn, a number of molecules will be produced, and these relay molecules can be seen being produced in a signal transduction pathway. So after the reception of signal, the transmission of the signal across the membrane into the cytosol and initiation of biochemical reaction, this is called transduction of the signal. Then last comes response. Say for example, it is a hormone that initiates the, you can say, cell growth and multiplication in this particular cell. So these relay molecules will now activate that biochemical pathway, and that cell will undergo division. So this is called response. That is, according to the message which is received on the plasma membrane, after the transduction of that message, response, or what biochemical, you can say, after the initiation of a biochemical reaction, what response is being initiated in that cell, that is the last part. So this is called activation of the cellular response. So this is how reception, transduction, and response, they work in signal transduction pathway. So here in this figure, you can see an example of signal transduction, where again here you'll see the reception part, the transduction, and the response part. So this is the extracellular side, this is plasma membrane, and this is the cell interior. During the reception, say a growth factor goes and binds to the receptor present on the plasma membrane. Of course, its intracellular domain initiates a phosphorylation cascade in the cytoplasm that produces a transcription factor. This part is representing the nucleus, which travels through the nuclear pore, and you can say it activates the transcription factor, which was initially inactive, that goes and binds to DNA and produces the mRNA that is required for that particular response. So here you can see how a signaling molecule from a distant cell can act on the DNA of another cell, at DNA level of another cell. So this is how signal transduction works, and this is the significance of cell-to-cell signaling in living systems. Thank you so much.